Hello everyone, thank you for coming to attend my presentation. My name is Tessema Kassaw. I am from Dr. Pierce Lab at Colorado State University. Before I start my talk, I want to thank Andrew Patton, a graduate student, and my PI Dr. Pierce. This project is funded by US Department of Energy, Office of Science Division. Today, I want to discuss my work on synthetic promoters for controlled gene expression in the module diatom Phaeodactylum. First, let's briefly see why diatoms are suitable choices for a synthetic biology application. As you all know, diatoms are a diverse group of organisms with large untapped biotechnological potential. Number of genetic engineering tools are rapidly increasing. Sequence information of multiple diatoms and metabolic models are available. However, a standard and modular genetic device or components that advance synthetic biology in other model organisms are limited in diatoms. Towards this end, we designed and evaluated 6 inducible expression systems that has been proven effective in other eukaryotic organisms. Due to time constraint, I will only focus on one of the system, the beta-stridal inducible expression system. Beta-stridal inducible expression system is tightly regulated and exploited in multiple organisms. We adapt this system from plants. As any other chemical inducible expression system, the beta-stridal inducible expression system has 2 transcriptional units. One, a constitutive promoter driving a transcript factor. 2, an inducible promoter driving a reporter protein or a gene of interest. What are the desirable properties of a synthetic inducible promoter? First, a synthetic inducible promoter should have negligible basal activity, and it has to be tunable in a dose and time dependent manner, and the effect has to be reversible. In the following few slides, I'm going to demonstrate the successful implementation of our inducible system, fulfilling all these desirable properties from the epizone. First, we evaluated the dose-dependent dynamics of the inducible system. And for this, we use a longish factor 2 constitutive promoter to drive the synthetic transcription factor. For simplicity, we include 1 wild type, 1 negative control, and 1 beta-stridal inducible exconjugant. We measured fluorescent intensity, which is depicted in the y-axis using a flow cytometer. This is 24-hour post-induction, and we test 6 inducer concentration levels. As you can see, in the absence of an inducer, our system showed negligible basal activity, which is equivalent to a background signal coming from wild type or negative control, which demonstrating no promoter leakness. However, with increasing concentration of an inducer, we observed an increasing fluorescent intensity. This is in a dose-dependent manner. It shows that our system is tunable in a dose-dependent manner. Next, we sought to determine the temporal characteristics of the system. We used the same strains for this purpose, and we determined the optimal inducer concentration to be 1 macromolar beta-stridal. And I use this concentration throughout my presentation. We measured background fluorescent intensity before inducer application and assigned as the 0 time point. We treat cells with 1 macromolar beta-stridal and measured the fluorescent data for a 50-hour period. It's a total of 9 time points. As you can see, strains expressing our inducible expression system showed a gradual increase in fluorescent intensity over time and attained a steady state level of expression within 24-hour period. and they maintain that level of expression throughout the experimental period. This shows that our inducible system is attainable in a time-dependent manner. However, we think that the messenger RNA induction or activation may be detected much faster than what we see in this fluorescent data. For this, we use a droplet digital PCR and measure the kinetics of messenger RNA production over time. And our assumption was correct. We observed significant accumulation of messenger RNA within minutes of industrial application and saturates after 6 hours, which is much faster than the one observed in the fluorescent data, 20 hours faster to be exact. This shows that our system is robust and tunable in a time-dependent manner, both at translational and transcriptional level. One of the requirements for a chemical regulator expression system is reversibility. To demonstrate the reversibility of our system, we treat cells with 1 macromolar beta-steroidol and remove the inducer by washing 48 hours after induction. And we assign that time point, the 0 time point. And we measured a fluorescent intensity over time. As you can see, the X-conjugates expressing our inducible expression system showed a gradual decrease in fluorescent intensity and reached less than half of the maximum fluorescent intensity within 24-hour period. And then almost showed undetectable fluorescent level 2 days after inducer withdrawal. This showed that our system is reversible. In conclusion, we developed at least 2 ultra-sensitive, tunable, reversible chemical inducible expression system in Phaeodactylum. To our knowledge, this is the first successful synthetic promoter reported for control gene expression in microalgae. We believe that the system described here will expand the molecular toolkits to facilitate synthetic biology first in algae. Thank you for your attention, and I'll be happy to answer your question. Thank you.